Hey everyone, welcome to Regenmedglobal The Podcast, where we bring you the top doctors and researchers in the world leading the newest breakthroughs in medicine and research. My name is Shine. What's the major breakthrough that we're talking about today? Today we're going to be talking about the use of nanoparticles to reduce neuroinflammation that's been done in a study of mice. Involved in this research, we have Owen Fenton, assistant professor at University of North Carolina School of Pharmacy. Owen, thank you so much for joining us.
Thanks so much for having me.
Of course, before we get started, do you mind maybe just telling us a little bit about what you do?
Sure, absolutely. So my name's Owen Fenton. I am an assistant professor at the University of North Carolina at Chapel Hill in the Eshelman School of Pharmacy. And my laboratory focuses on the development of different types of genetic medicines, with a particular interest in some areas like Alzheimer's disease, which are characterized by different forms of neuroinflammation. So what we do is we come up with new strategies to maybe treat diseases that are difficult or impossible to treat with other types of medications.
Awesome. Before we get started, can you also give us a little introduction as to what nanoparticles are?
Yeah, absolutely. It's a great question. So nanoparticles are one type of vehicle that we can use to make medicines better. So all sorts of medicines have been developed through time, and one challenge with them is it can be difficult to control the dose, the location, and the amount of time that a medicine is actually available to a disease site inside of the body. What nanoparticles do is they allow us as engineers and scientists to come up with controlled ways to deliver drugs better to certain areas inside of the body. And this is really important because many diseases need to actually be treated by controlling the specific time or dose of a given therapy in a location without causing off-target effects. And so nanomedicine is basically just a fancy way of coming up with better ways to make people feel better with different types of medications.
Awesome. Are nanoparticles used at all right now for any treatments, or is it still kind of at the research stage?
Yeah, another really great question. So nanomedicines have a really long and storied history, and perhaps the kind of history that's most relevant to, I guess, what we'll talk about in a little bit is actually the clinical successes of the Moderna and Pfizer-BioNTech COVID-19 mRNA vaccines. So these mRNA vaccines that were widely deployed throughout the COVID pandemic actually leverage nanoparticle-based technologies. And what these are kind of little bubbles of fat almost that are able to protect and deliver messenger RNAs into the body and train our immune system to protect people against harmful pathogens like the COVID virus. And so nanoparticles have a really long and storied history. They definitely help localize specific types of drugs into different compartments in vivo. And for nucleic acid-based therapies like messenger RNA vaccines, or in our case in this study, siRNA or short interfering RNA-based therapies, they're really essential to make this medicine function. For example, if you deliver or use a nucleic acid-based drug like messenger RNA or siRNA without a carrier, its efficiency is significantly reduced. And that's because these nucleic acid drugs are very unstable. They have a really strong negative charge, which prevents their efficient delivery into cells of interest. And they also don't necessarily localize to the area of interest. And so what our nanoparticles do is they better deliver these drugs to a site of disease and ultimately make that medicine safer, more effective, and more targeted than if they were delivered without a nanoparticle.
With the study that you and your team did on the mice in regards to nanoparticles and neuroinflammation, can you give us an overview of that study and kind of what the goal was?
Absolutely. And so I'd also like to just mention, a number of really key and excellent players in this. So this study actually highlights a lot of interdisciplinary work. There's several key players, and I won't have time to mention them all, but Jason Anderson is a really fantastic PhD student who received his PhD at MIT. We actually both knew each other because I did my PhD at MIT as well. There was a postdoctoral study named William Malvinius, who was also incredibly helpful, who works at MIT as well. And then myself, Robert Langer, and Liwei Tsai are all co-senior authors on this manuscript. Bob is a major expert in the drug delivery sciences and is really essential towards creating nucleic acid therapies. Professor Liwei Tsai is the director of the Picower Institute at MIT and focuses really heavily in a lot of different areas, including things like Alzheimer's disease. Similarly, I'm a professor at the University of North Carolina in the pharmacy department. I received my PhD from MIT and also my postdoctoral studies there as well. Had the opportunity to train with Bob directly there and other fantastic faculty members like Dan Anderson. And then also was able to start collaboration on this work with Liwei Tsai, which is what we've continued into this study. Now, building on that, the overarching goal of this study was to come up with new types of therapies and explore potential therapeutic avenues to better treat things like Alzheimer's disease. So, as many people know, Alzheimer's disease is a progressive neurodegenerative disease. It's characterized by things like memory loss, cognitive decline, and ultimately what it can do is lead to an inability to perform kind of daily tasks. People will forget things. Can be really kind of a psychologically and emotionally damaging disease for many people around because there aren't a lot of great therapies that we can do to reverse this. And so while the person is still alive, they may actually not be fully aware of their surroundings at times or maybe struggling with kind of different tasks. And so the overall goal of this study was actually to think about leveraging kind of our expertise in nanomedicine as well as the team's expertise in neurodegeneration to explore nucleic acid-based therapies for Alzheimer's. What that means basically is Alzheimer's is a disease that's characterized by a process called neuroinflammation. So typically we think about inflammation being throughout the body. Generally, it can be considered a negative thing for certain types of disease. What we're trying to do basically here is actually turn down the dial of inflammation inside of an Alzheimer's patient, in this case it's in a mouse, to better study and treat Alzheimer's disease.
Awesome. What were some of the findings from this just taking this approach?
Yeah, so there's, I would say, a couple of key findings that were involved here. So the first, as I mentioned, is that Alzheimer's disease is a disease of neuroinflammation. And what this means is that there are cells inside of our brain, especially microglia, which is kind of the primary resident immune cell inside of our brain. In Alzheimer's disease, there can be upregulation of certain genetic factors that will correlate with worse disease progression. One of the main ones we look at here is what's called a transcription factor, which is a fancy way of saying kind of a molecule that helps to control a lot of processes going on inside of the brain. And what we do is we look at transcription factors such as PU.1, which are often upregulated in the case of Alzheimer's disease. What we do basically is we create new types of therapies where we can reduce those levels, especially within microglia, and use that reduction in neuroinflammation to kind of try to reverse or better care for Alzheimer's disease patients. In this case, again, this whole study is done in mice.
Awesome. Now, I mean, just with all of this, is there, why do you think this study is so important? I mean, studying in neuroinflammation, Alzheimer's, what makes this study so unique compared to all the other maybe potential treatments or potential studies that have been done on Alzheimer's?
Yeah, so I think what makes this study particularly unique is really the synergy of the expertise that comes in here, ranging from really excellent nanoparticle engineering to really classic understanding of neurodegeneration, especially in the case of Alzheimer's disease. What makes this very unique is that we're kind of targeting a unique pathway, i.e. inflammation, and that kind of stands in contrast to some of the other therapies that are out there. So what we're looking at doing is targeting a unique pathway and leveraging very targeted, safe, and effective medication-based strategies to try to overturn some of those classically difficult or impossible to treat pathways inside of Alzheimer's disease.
Okay. Now, in terms of just the study on the mice, going back to that, were there any particular the physical benefits that you saw from the mice? Were there any particular symptoms or anything of that nature that you guys saw that improved within this experiment?
Yeah. So in this study, what we did is we analyzed basically mice that have chronic inflammation. We can induce this in different animal models. And what we did is we basically created an experiment where we either treated our mice with our kind of new medicines or didn't. And what we analyzed basically is a number of different functions of these mice. So one way to kind of analyze the behavior of a treatment for an Alzheimer's type model in mice is to actually see how far or how much they walk, what's their walking patterns. And we saw that actually these medicines are able to bring back function closer to kind of a better disease state when they're treated with our nanoparticle-based systems. Similarly, we can look at this in a targeted fashion. What I mean by that is we can actually correlate basically the decreases in inflammation with this by actually quantifying and looking at the specific gene that we're trying to downregulate. Similarly, what we were able to also identify here is actually these nanoparticles work better than commercial systems, highlighting kind of the need of these customizable, tunable nanoparticle platforms that are safe, targeted, and effective at delivering these really important genetic medicine payloads.
That's great, Owen. So, what's the current status of the study right now?
Yeah. So this study was actually just recently published in this really fantastic journal called Advanced Materials. It's a really excellent journal that is super interdisciplinary in nature, bringing together experts in nanomedicine, materials, therapy, things like Alzheimer's disease. And so if you're interested, this manuscript is actually published open access. So what that means is anyone can read it, and it has the results actually detailed online. Where we're going next is we have this really fantastic collaboration where we're continuing to explore even better versions of these types of nanomedicines and further kind of understanding and using these approaches to understand some of the complexities of the origins of disease. And so it's this really fantastic group of scientists that I consider myself very, very fortunate and privileged to have worked with and learned from. And so we're all kind of working together towards trying to create better therapies that can treat, progressive neurodegenerative diseases like Alzheimer's, and perhaps one day may also be valuable for treating other types of disease and better understanding it. So the ultimate goal of kind of almost all of our studies is to think about how can we create better medicines to help people. And that's kind of one of the essential goals and overall kind of metrics in this study.
This potential treatment that you guys have developed for Alzheimer's, do you see it potentially being available for humans someday?
I think it's always difficult to say, right? The pathway from going from mice to humans carries a lot of challenges and a lot of complexities. That being said, I think that, we're definitely on the right path, right? You have to start by showing that this can actually work in animal models, and then those can progress up. Now, I think that this team is really excellent and combines both translational expertise as well as nanomedicine and the appropriate kind of clinical endpoints as well, especially thinking with the expertise of, again, my co-senior authors, Li Wei Sai, who's the director of the Picower Institute and is a professor at MIT, and then Bob Langer as well, who's really been a fantastic mentor and is a professor at MIT as well as my lab, which focuses on different aspects of the nanoparticle chemistry. So while it's impossible to kind of predict very long term, I think that this technology is certainly on the right path towards creating better, more targeted, and safer therapies for the treatment of Alzheimer's disease.
Owen, this is all very valuable information and very insightful. Any other kind of final thoughts or anything else that you think would be important to share about this study or the direction of kind of where this is going with nanoparticles?
Yeah, no, I would just say, thank you so much for the opportunity to highlight our work. Our team is, again, consider myself very lucky to have worked with amazing co-authors and scientists that are curious, dedicated, and passionate to creating new types of disease therapies. If it's of interest, again, the manuscript, which details the study, is actually published on Live and it's open access. So you can actually go ahead and check it out for yourself and kind of think about some of the advances there. We're always happy and excited to hear kind of more insight into this and think about, how nanomedicines can be used to treat really complex and important diseases, especially in humans long term. So yeah, happy to chat further, and we super appreciate, or at least I super appreciate, and I'm sure my team would probably echo the same sentiments, the opportunity to highlight our work. So thank you so much for having us on the podcast. I very much appreciate it.
Owen, thank you so much. Truly a pleasure speaking with you and sharing all this information. This was great. Wishing you all the best. Thank you.
Pleasure is all ours. Thanks.